Section 10 of Gray's Anatomy Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ellie. Anatomy of the Human Body Part 5 by Henry Gray. The Mouse Part 3. Development of the teeth. In describing the development of the teeth, the mode of formation of the deciduous teeth must first be considered, and then that of the permanent series. Development of the deciduous teeth. The development of the deciduous teeth begins about the 6th week of fetal life as a thickening of the epithelium along the line of the future jaw, the thickening being due to a rapid multiplication of the more deeply situated epithelial cells. As the cells multiply, they extend into the subjacent mesoderm and thus form a richer strand of cells embedded in mesoderm. About the 7th week, a longitudinal splitting or cleavage of this strand of cells takes place, and it becomes divided into two strands. The separation begins in front and extends laterally, the process occupying 4 or 5 weeks. Of the two strands thus formed, the labial forms the labiodental lamina, while the other, the lingual, is the ridge of cells in connection with which the teeth, both deciduous and permanent, are developed. Hence it is known as the dental lamina or common dental germ. It forms a flat band of cells, which grows into the substance of the embryonic jaw, at first horizontally inward and then, as the teeth develop, vertically, i.e. upward in the upper jaw and downward in the lower jaw. While still maintaining a horizontal direction, it has two edges, an attached edge, continuous with the epithelium lining the mouth, and a free edge, projecting inward and embedded in the mesodermal tissue of the embryonic jaw. Along its line of attachment to the buccal epithelium is a shallow groove, the dental furrow. About the 9th week the dental lamina begins to develop enlargements along its free border. These are 10 in number in each jaw, and each corresponds with a future deciduous tooth. They consist of masses of epithelial cells, and the cells of the deeper part, that is, the part farthest from the margin of the jaw, increase rapidly and spread out in all directions. Each mass thus comes to assume a club shape, connected with the general epithelial lining of the mouth by a narrow neck, embraced by mesoderm. They are now known as special dental germs. After a time the lower expanded portion inclines outward, so as to form an angle with the superficial constricted portion, which is sometimes known as the neck of the special dental germ. About the 10th week, the mesodermal tissue beneath the special dental germs becomes differentiated into papillae. These grow upward and come in contact with the epithelial cells of the special dental germs, which become folded over them like a hood or cap. There is then at this stage a papilla or papillae, which has already begun to assume somewhat the shape of the crown of the future tooth, and from which the dentin and pulp of the tooth are formed, surmounted by a dome or cap of epithelial cells, from which the enamel is derived. In the meantime, while these changes have been going on, the dental lamina has been extending backward behind the special dental germ corresponding with the second deciduous molar tooth, and at about the 17th week it presents an enlargement, the special dental germ for the first permanent molar, soon followed by the formation of a pillar in the mesodermal tissue for the same tooth. This is followed about the 6 months after birth by a further extension backward of the dental lamina, with the formation of another enlargement and its corresponding papilla for the second molar. And finally the process is repeated for a third time, its papilla appearing about the 5th year of life. After the formation of the special dental germs, the dental lamina undergoes atrophic changes and becomes cribriform, except on the lingual and lateral aspects of each of the special dental germs. After the formation of the special dental germs, the dental lamina undergoes atrophic changes and becomes cribriform, except on the lingual and lateral aspects of each of the special germs of the temporary teeth, where it undergoes a local thickening forming the special dental germ of each of the successional permanent teeth, the 10 anterior ones in each jaw. Here the same process goes on as has been described in connection with those of the deciduous teeth. That is, they recede into the substance of the gum behind the germs of the deciduous teeth. As they recede they become club-shaped, form expansions at their distal extremities, and finally meet papillae, which have been formed in the mesoderm just in the same manner as was the case in the deciduous teeth. The apex of each papilla indents the dental germ which encloses it, and forming a cap for it, becomes converted into enamel, while the papilla forms the dentin and pulp of the permanent tooth. The special dental germs consist at first of rounded or polyhedral epithelial cells. After the formation of the papillae, these cells undergo a differentiation into three layers. Those which are in immediate contact with the papilla become elongated and form a layer of well-marked columnar epithelium coating the papilla. They are the cells which form the enamel fibers and are therefore termed enamel cells or odontoblasts. The cells of the outer layer of the special dental germs, which are in contact with the inner surface of the dental sac, presently to be described, are much shorter, cubical in form, and are named the external enamel epithelium. All the intermediate round cells of the dental germ between these two layers undergo a peculiar change. They become stellate in shape and develop processes which unite to form a network into which fluid is secreted. This has the appearance of a jelly, and to it the name enamel pulp is given. This transformed special dental germ is now known under the name of enamel organ. While these changes are going on, a sac is formed around each enamel organ from the surrounding mesodermal tissue. This is known as the dental sac, and is a vascular membrane of connective tissue. It grows up from below and thus encloses the whole tooth germ. As it grows, it causes the neck of the enamel organ to atrophy and disappear, so that all communication between the enamel organ and the superficial epithelium is cut off. At this stage there are vascular papillae, surmounted by caps of epithelial cells, the whole being surrounded by membranous sacs. Formation of enamel. The enamel is formed exclusively from the enamel cells or odontoblasts of the special dental germs, either by direct calcification of the columnar cells, which become elongated into hexagonal rods of the enamel, or, as is more generally believed, as a secretion from the odontoblasts, within which calcareous matter is subsequently deposited. The process begins at the apex of each cusp, at the ends of the enamel cells in contact with the dental papilla. Here a fine globular deposit takes place, being apparently shed from the end of the odontoblasts. It is known by the name of enamel droplet, and resembles keratin in its resistance to the action of mineral acids. This droplet then becomes fibrous and calcifies, and forms the first layer of the enamel. A second droplet now appears and calcifies, and so on. Successive droplets of keratin-like material are shed from the odontoblasts, and form successive layers of enamel, the odontoblasts gradually receding as each layer is produced, until at the termination of the process they have almost disappeared. The intermediate cells of the enamel pulp atrophy and disappear, so that the newly formed calcified material and the external enamel epithelium come into opposition. This latter layer, however, soon disappears on the emergence of the tooth beyond the gum. After its disappearance the crown of the tooth is still covered by a distinct membrane, which persists for some time. This is known as the cuticular dentis, or Nasmyth's membrane, and is believed to be the last formed layer of enamel derived from the odontoblasts, which has not yet become calcified. It forms a horny layer, which may be separated from the subjacent calcified mass by the action of strong acids. It is marked by the hexagonal impressions of the enamel prisms, and when stained by nitrate of silver shows the characteristic appearance of epithelium. Formation of dentin. While these changes are taking place in the epithelium to form the enamel, contemporaneous changes occurring in the differentiated mesoderm of the dental papillae result in the formation of the dentin. As before stated, the first germs of the dentin are the papillae, corresponding in number to the teeth, formed from the soft mesodermal tissue, which bounds the depressions containing the special enamel germs. The papillae grow upward into the enamel germs and become covered by them, both being enclosed in a vascular connective tissue, the dental sac, in the manner above described. Each papilla then constitutes the formative pulp, from which the dentin and permanent pulp are developed. It consists of rounded cells and is very vascular, and soon begins to assume the shape of a future tooth. The next step is the appearance of the odontoblasts, which have a relation to the development of the teeth similar to that of the osteoblasts to the formation of the bone. They are formed from the cells of the periphery of the papilla, that is to say, from the cells in immediate contact with the odontoblasts of the special dental germ. These cells become elongated, one end of the elongated cell resting against the epithelium of the special dental germs, the other being tapered and often branched. By the direct transformation of the peripheral ends of these cells, or by a secretion from them, a layer of uncalcified matrix, prodentin, is formed, which caps the cusp, or cusps, if there are more than one, of the papillae. This matrix becomes fibrillated, and in it islets of calcification make their appearance, and coalescing gives rise to a continuous layer of calcified material which covers each cusp and constitutes the first layer of dentin. the odontoblasts having